Good afternoon to everyone joining us today. I welcome all of you to the second half of this online program. We are honored to have participants from various institutions and locations, and we appreciate you taking the time to be with us. Few guidelines for smooth participation. Please keep your microphones on mute during the presentation. We will address the questions during the question answer session at the end of each lecture. You can write to us in the chat box or can directly ask question to the speaker during the question answer session. So today we are privileged to have with us Professor K. Sanatombi, Department of Biotechnology, Manipur University, Imphal. With expertise in plant biotechnology, molecular biology, and in vitro production of secondary metabolites, she obtained her MSc in botany from Punjab University and earned her PhD from Manipur University. Over the years, she has established herself as a leading academician and researcher in the field of plant sciences, particularly in exploring the rich plant biodiversity of Northeast India through modern biotechnological tools. Her research emphasizes molecular profiling, genetic diversity analysis, and the enhancement of bioactive compounds from medicinal and aromatic plants. Her pioneering work includes studies on in vitro culture systems for Capsicum chinense, Zingiber montanum, and Strobilanthus cusia, aimed at optimizing metabolites production and conserving valuable plant species. A prolific researcher, she has published numerous papers in reputed international and national journals and has guided several PhD and postgraduate students. Beyond her laboratory work, she has contributed to higher education through innovative teaching and as a course coordinator for national level online MOOCs on the SWAYAM platform. In recognition of her academic excellence and leadership, Professor Sanatombi has received various honors, including the Women Leadership in STEM program. She is also an active member of several scientific societies, including the Indian Science Congress Association and the Society of Biological Chemists. So without further delay, let us begin. But before that, I would like to request Madam to give us some time at the end of her presentation so that we can take questions from the audience and can end the session before 3 p.m. so that our next speaker can start sharp at 3:30 p.m. Thank you, Madam. So I request Professor Sanatombi to take over the session.
Thank you for
Madam.
Thank you for a kind introduction. And let me start my presentation now.
Yes, ma'am.
So, is my screen visible?
Yes.
Yes, ma'am. Your screen is visible and you're audible also. Yes.
Okay, thank you so much.
Thank you, ma'am.
So, a very good afternoon, everyone. For this online refresher course on bioscience, bioengineering, and nanotechnology, organized by MMTC, University of Calcutta, and Maulana Azad College. Today, the topic of my talk is nanoelicitation, harnessing nanoparticles to stimulate secondary metabolism in plants. Now, most of the participants may be from different backgrounds, so I would like to start with a brief introduction of plant secondary metabolites. This plant secondary metabolites, as the name suggests, they are compounds which are produced as a result of secondary metabolism in plants. The primary metabolites, which play a key role in the growth and development of plants. Now, in addition to the primary metabolites, the plants, they also produce a lot of secondary metabolites, and these compounds are usually produced in small quantities in plants. Unlike these metabolites are not actively or are not directly involved in the growth and development, but they play other key roles in the plants, like in defense and adaptation to stresses. They help the plants to protect themselves from various biotic and abiotic stresses. So these compounds, their primary role is to protect the plants from stresses and for adaptation. But in addition to their role in plants, these compounds are useful to plants, humans, and many of these compounds find uses in industries. So, in plants, they play various roles like they may act against the pathogens, herbivores, environmental stresses, or they help in attracting pollinators or in symbiotic association. So we will see about the synthesis and groups of these compounds, but the idea is that these compounds are very much useful to uh, these compounds are useful to human beings in different purposes. Many of these compounds are used as pharmaceutical drugs or as nutraceuticals or in industries as anti-cancer compounds, antimicrobials, or antioxidant agents. So, uh, in the industrial application for these compounds includes use in food flavoring, like the some common examples are the pigments, carotenoids, or other compounds like the curcumin from turmeric, etc. And we also have many compounds which are used in textile productions or dyeings or as detergents and cosmetics. So let us see the secondary metabolite biosynthesis pathways in plants. Now, the primary carbon metabolism, starting with photosynthesis, combining carbon dioxide and water. Now, this undergoes the primary carbon metabolism, and many compounds or intermediate compounds which are produced or which are involved in the primary carbon metabolism, they enter the secondary metabolism pathways inside the plant. So some of the intermediate compounds like the erythrose 4-phosphate, phosphoenol pyruvate, pyruvate or 3-phosphoglycerate. Now, these are some of the compounds which enter different secondary metabolism pathways like the cyclic acid pathway, the malonic acid pathway, the mevalonic acid pathway, and the MEP pathway. So, through these different pathways, these are not exactly the uh, primary metabolism pathways like the glycolysis or TCA cycles, but these are additional secondary metabolism pathways that operate inside the plants. So, through these pathways, they give rise to a lot of secondary metabolites. So, for example, the cyclic acid pathway, they produce aromatic amino acids, which gives rise to nitrogen containing secondary metabolites. Similarly, from Succinic acid pathways or malonic acid pathways, they contribute to the synthesis of a large group of compounds known as the phenolic compounds. While other group of compounds which is produced as a result of the secondary metabolism are the terpenes. So, if we see the secondary metabolites, the different groups of secondary metabolites, simply, or we can group them in common groups like the terpenes, phenolics, nitrogen containing compounds, or the sulfur containing compounds. These are some examples. The terpenes includes a large group of compounds which are grouped depending upon the terpenoid unit, like the monoterpenes, sesquiterpenes, diterpenes, sesquiterpenes, triterpenes, etc. So these are some of the compounds. So we see the common example lycopene here. So lycopene is a type of terpene. Then the next group of compounds are the phenolics. So these phenolics are also a large group of compound, and we have different examples including the tannins, flavonoids, lignins, etc. Then the next are the nitrogen containing compounds, among which the most important groups are the alkaloids, like the alkaloids from morphine alkaloids or cocaine alkaloids. And then another example, common example, includes the pungent compounds in capsicum or the capsaicin. Then the next last group includes the sulfur containing compounds, which includes, these are common in like glutathione, glucosinolates, phytoalexins. So alanine, this is common in alliums like the garlic or onions. And then sulfur containing compounds are also very common in the cruciferous family, that crucifery family, that is the mustard family. Now coming to elicitation. Now, elicitation is the phenomenon where we trigger the synthesis of these metabolites. So, in plants, these compounds are usually produced in very small quantities, but elicitation is the phenomenon by which we can stimulate or trigger the synthesis, increase the synthesis of these compounds. So, there are different types of elicitors which stimulate the or which enhance the synthesis of these compounds in plants. For example, depending upon the source, the elicitors may be either biotic or abiotic. So biotic elicitors includes microbes, fungal or bacterial extracts, or polysaccharides or compounds. These are compounds which imitate the biotic sources, the pathogens, etc., and they stimulate the synthesis of the secondary metabolites. Similarly, different abiotic elicitors also stimulate the synthesis of secondary metabolites. Some examples include ultraviolet radiation, metals, or hormones like methyl jasmonate and salicylic acid. So these abiotic factors, they also stimulate the synthesis of secondary metabolites. So why we are talking about elicitation, or what is the importance of elicitation, is that when we use elicitors, we can increase the synthesis of this important secondary metabolites in plants. So when the synthesis of the secondary metabolites increases in the plants, now they may enhance stress tolerance in plants, or they may increase the productivity of the secondary important secondary metabolites, which can be used for different purposes. So whether in plants, in vivo or in vitro in tissue culture condition, now if we can elicit the production of secondary metabolites, we can increase the yield of the secondary metabolites. Now coming to nanoparticles. Now we will there are different types of nanoparticles, and nanoparticles differ on they are small particles whose size vary from about 1 to 100 nanometers. So different types of nanomaterials or nanoparticles have been used as elicitors. Now we will see about the different examples later on, but here let us see some different types of nanoparticles which can be used for elicitation. So we have solid core mesoporous nanoparticles. We may use polymeric or dendrimers, nanorods, mesoporous. These are different types of nanoparticles, but the examples and their use depends upon our choice or purpose. So here from this table, we see that there are different nanoparticles which have been used in nanoelicitation. Means nanoparticles which have been used to enhance the production of secondary metabolites in plants. So the first example includes the metal nanoparticles. So this metal nanoparticles includes nanoparticles like the silver nanoparticles, gold nanoparticles, copper nanoparticles, or cobalt nanoparticles. So this metallic nanoparticles, they usually have high surface reactivity, and they are capable of redox reaction, inducing redox reactions inside the plants. And another important feature is that their size and shape can be modified or tuned depending upon our requirement. Now, the next type of nanoparticles commonly used are the metal oxide nanoparticles. Now, this metal oxide nanoparticles like the zinc oxide nanoparticles, copper oxide nanoparticles, titanium oxide nanoparticles, or iron oxide nanoparticles, these are another group of nanoparticles which are very stable, and they have photocatalytic properties. So these are also commonly used for elicitation of secondary metabolism in plants. Then another type of nanoparticles which can be used for nanoelicitation are the magnetic nanoparticles, like the iron oxide nanoparticles. Now, the one important property of such nanoparticles is that they exhibit magnetism. So because of this, they are easily recoverable and they are easy to handle. And in addition to that, they also have very good biocompatibility, means they can be used in without much toxicity. Then another type or group of nanoparticles are silica and non-metal nanoparticles, like silicon dioxide nanoparticles and alumina nanoparticles. Now, these are usually porous and inert with high surface area, and they can serve as good carriers for different biologicals, as well as they themselves can be used directly as in nanoelicitation. Then another important group of nanoparticles are the carbon-based one. So in this, we have examples like the single-wall carbon nanotubes or the multi-wall carbon nanotubes or graphene oxide, fullerenes, etc. Now, such type of carbon-based nanomaterials, they have high mechanical strength and unique electronic properties. And they are also easily functionalized, means that we can easily add different functional groups to these nanoparticles. The next are the semi-metal and chalcogen nanoparticles. Like in this, we have examples like the selenium nanoparticles, tellurium nanoparticles, or sulfur nanoparticles. Now, these nanoparticles also act as micronutrient sources which are useful for nutrition in plants, and they also can be used as efficient nanoparticles for elicitation. The other groups includes the hybrid or composite nanoparticles, where we can combine different nanoparticles like silver and silicon dioxide, titanium dioxide with perlite, or hybrids of polymers and metals. So when we use such type of hybrid or composite nanoparticles, now we can control the release of the metal ions, or we can modify or tune the surface chemistry of the nanomaterials also. Then the next type are the biopolymer coated nanoparticles. Now, different biopolymers like chitosan or alginate or PLGA, that is polylactic glycolic acid coated nanoparticles, have also been used for nanoelicitation. Now, this coating the nanoparticles with these polymers, they increase the stability of the nanoparticles, and they also allow the controlled release of metal ions or any other particles which are being coated with these biopolymers. Then the last other group of nanoparticles which can be used in nanoelicitation are the green synthesized nanoparticles. So such type of nanoparticles are the nanoparticles which are synthesized using plant or microbial extract. So different plant extracts can be combined with silver or zinc oxide and nano or iron oxide, and nanoparticles may be generated. So another important feature of such nanoparticles is that these are usually eco-friendly, and these nanoparticles are also commonly used for nanoelicitation. So why we are discussing about nanoelicitation, or what is the importance of nanoelicitation, is that by using the different nanoparticles, we can manipulate the secondary metabolism in plants, and we can enhance the production of the secondary metabolites. So uh, basically what is happening is that these nanoparticles can be used as abiotic elicitors, and we can trigger the synthesis of the important metabolites in plants, which can be used for different purposes. So conventionally, in conventional agriculture or conventional production, hormones like the methyl jasmonate or salicylic acid, they have been used as elicitors, or sometimes abiotic compounds like chitosan have also been used as elicitors to increase the production of secondary metabolites. Now, when we use nanoparticles in elicitation, now the nanoparticles can be applied in different ways. So the most common, there are three common methods by which we can apply the nanoparticles. The first one is the foliar application, means we can apply the nanoparticle directly on the aerial or leaves of the plants by spraying as nanoparticle sprays. So this is one common method of application. Then the second one is soil addition or seed priming. That means we can either add the nanoparticles directly in the soil, or we can coat the seed with the nanoparticles. So these are another type of application. And then in plant tissue culture, when we study the secondary metabolite production under in vitro condition, we can directly inject or put it in the culture medium. So these are some common methods of application of uh, nanoparticles in plants, and the uptake and translocation of these nanoparticles depend on the type, size, concentration, or the mode of application. So let us see how these nanoparticles are applied and how their uptake occurs inside the plants. So, as discussed, the nanoparticles can be applied by spraying directly on the plants as foliar sprays, or we can apply it in the soil for uptake through the roots of the plants. While in case of plant cell culture or tissue cultures, we can directly add the nanoparticles in the medium from which the cell cultures or the tissues, they can take up the nanoparticles. So when the nanoparticles comes in contact with the plant, now their entry inside the plant cells depends upon the type of cells they encounter. In the case of roots, when the nanoparticles are applied to the roots, the nanoparticles usually enter the plants through the root hairs. So they enter the plants through the root hairs, and then they move through the different layers of the root tissue, that is starting from the epidermis, then the cortex, then the endodermis, then finally they enter the conducting tissue, that is the xylem and phloem. So when they enter the once they enter the conducting tissue, then they can be transported to all parts of the plant. Well, when the application is through the foliar mode, most of the time what happens is that depending upon the size of the nanoparticles, if the nanoparticle size is small, now many times they can directly enter the leaves through the stomatal opening. So once they enter the leaves through the stomatal opening, then they can enter the rest part of the leaves, and from which they can be transported to other parts of the plant. Now, the entry into the plant cells may occur through different pathways. Means sometimes the nanoparticles can enter inside the cell by endocytosis. Means when the nanoparticles comes in contact with the membrane of the plant cell, they may be engulfed through a vesicle, and by endocytosis they may enter inside the cell. Then another pathway for entering is through carrier proteins. So different transport or carrier proteins located in the plasma membrane of the plant cells, they may these nanoparticles may be may attach to these carrier proteins. Now some common carrier proteins include the aqua protein or water transport protein present in the plant cells, and there are also other proteins like the ion channels or other proteins which may also help in transporting these nanoparticles. So they may also enter inside the plant cells through the carrier proteins. Then the next one is that some nanoparticles may directly rupture the membrane of the plant cells. So when they come in contact with the plant cells, they may form pores on the plant plasma membrane, and they may directly enter the plant cells. So these are some of the common methods or processes by which nanoparticles enter inside the plant cells. Then, once they enter inside the plant cells, now their transport inside the plants may occur through two pathways, that is, symplastic or apoplastic. Now, this we can explain the apoplastic pathway and the symplastic pathway here. So, the difference between the apoplastic and symplastic pathway is that in the apoplastic pathway, the nanoparticles they enter inside the plant, and then they move through the plant tissue in spaces like between the cell walls or middle lamella, like this. Means they they are absorbed through the plant cell wall, and then they move through the cell walls and the middle lamella, means the tissue which connects the adjacent cells of the plants. So they move through these areas in the adjoining areas of the cell wall, and they move through these areas like this. So this is the apoplastic pathway. While in symplastic pathway, what happens is that the nanoparticles they enter inside the plant cells, and they move through the connections inside the plant cells. So we have connections, the plant cytoplasm cytoplasms, the plant cells, the plant cells are connected by a structure known as plasmodesmata. So through these plasmodesmatal connections, the nanoparticles move from one cell to another, and they move through the cytoplasm, and we call this type of transport the symplastic pathway. So the pathway which involves the cell wall and the middle lamella is the apoplastic, while if they are transported through the cytoplasm through the uh, plasmodesmatal connections, we call it the symplastic pathway. So this is how they are uptake from the soil or leaves, and they are transported inside the plants. So this this is how I have already explained. So one method to how they enter the cell inside the cell is by either by endocytosis or by forming pores directly on the surface of the plasma membrane of the plant cells, or this is how they move through the plasmodesmatal connection. Elicitors means abiotic.
I think there are some internet issues, so please bear with this and let Madam come back. Madam, Professor K. Sanatombi, are we audible? Madam, your video has completely freezed. We are unable to hear anything. I think there are some technical glitch, so just be with us. Hopefully, Madam will join soon. Please stay with us. Hopefully, Madam will join soon. There is a technical glitch. We are unable to contact Madam, but hopefully soon she will join. The coordinator is trying to contact with Professor Sana Tombi. Hopefully she will join soon. Please stay. Yeah, madam has joined. Madam has joined, yeah.
Okay, thank you. Sorry.
No, no issues, madam. It
Okay,
happens. No problem at all.
I'm sorry. I'm, uh, I'm sorry, I lost the connectivity.
No, yeah, madam, we understand. It's absolutely no
So
problem.
let
You can
me
start, madam. If possible, you can start the presentation.
restart this one presentation. You may please tell me from. So please tell me. Yes, I'm starting. Please tell me where I.
Yeah, yeah, it was. No, ma'am, it's not visible. You are visible, but your presentation is not visible.
Presentation. Okay. Just give me some.
Yes, ma'am. Yes, ma'am.
Mhm. Yes, let me share the screen again. Share screen.
Yes, ma'am. Now the presentation is visible. Yes,
Yes.
ma'am.
Hi. So please tell me where we lost connection.
Ma'am, you were at apoplastic and symplastic pathway.
Okay, apoplastic and symplastic pathway. Okay.
Yes, ma'am.
So, uh,
One only, I think.
okay. So, is this the slide where we?
Yes, ma'am. This slide.
Yes, madam. Yes.
Yes, ma'am.
Okay,
This
okay,
is the slide.
okay. Thank you so much. Now, let me. I think there is some problem here. Yes. So let me. So, so let us continue with the. Have we? Did you miss this one? Did we miss this one?
No, madam. I think this was said.
I
If the
mean,
participant can confirm.
Yes.
We have not seen this slide, madam.
No.
okay. Then I will.
First time
Then
we are
we
seeing
will.
this slide.
Okay. Then we will continue with this one. So after entering the cells, after uptake by the plants through different ways, now the nanoparticles, they are transported either through the symplastic route or the apoplastic route. Then after entering inside the cell, how metabolites. That is the molecular mechanism involved in nanoelicitation. Now, how the secondary metabolites induce the synthesis of metabolites is that now the nanoparticles are external agents we are applying to the plants. So the plant cells, they perceive the nanoparticles as strays or damage signals, just like the attack by the pathogens or the herbivores. So when these nanoparticles are perceived by the plants as strays, now they induce a chain of molecular events. Now, one key events include the hormonal crosstalk. That means there are specific hormonal pathways inside the plants, like the salicylic acid pathway or the jasmonic acid pathway, which are involved in response to pathogens or herbivores in the plants. So this hormonal pathways, what they do in normally in the plants is that when the pathogens or herbivores attack the plant cells, now these pathways, they induce the synthesis of secondary metabolites which help in deterring or which are toxic to the pathogens or herbivores. So these compounds, actually these are the compounds which are useful as secondary metabolites for different purposes. Now, in addition to the hormonal crosstalk, now the entry of the nanoparticles inside the plant cells, they may also induce the production of reactive oxygen species. So when these reactive oxygen species are produced inside the plants, they may activate different signaling pathways, including the mitogen activated protein kinase cascades. So the activation of such pathways lead to the different activation of different gene expression processes which are involved in expressing the enhancing the expression of gene or enzymes involved in secondary metabolism pathway. So the reactive oxygen species signaling or ROS production is another important aspect of nanoparticle exposure in plants. And as the reactive oxygen species are produced in response to nanoparticles, the plants, they may also produce antioxidant enzymes like the superoxide dismutase or ascorbate peroxidase and the catalases. So this production of these antioxidant enzymes, they are produced as a response to reactive oxygen species inside the plants. Then as we the signaling transduction pathway commonly involves binding of the nanoparticles to the cell surface receptors, which may activate certain downstream molecules like the G protein cascades, and they may lead to calcium signaling. So calcium signaling is another important pathway for molecular mechanism, and this increase in the calcium ion or concentration inside the cell, they may trigger the protein kinases including the MAPKs. So once these are triggered, phosphorylation will take place, and other secondary molecules like the inositol triphosphate or DAZ, they may further amplify the calcium concentration and the ROS signaling. So let us see this in this slide. So here, how the nanoparticle induce the synthesis of secondary metabolites in the plants. Now, let us see that the nanoparticles, when they come in contact with the plant cells, now here they are acting as the elicitors, means how they induce the synthesis of secondary metabolite starts with their interaction with the receptors on the membranes. So once they come in contact with the membrane, or once they enter inside
अरविंद
the
कहाँ हो
cell,
तेरा
Participant, please switch off your microphone. OP Singh, please switch off your microphone. You are interrupting the
ओके
session.
ओके
OP Singh.
so the nanoparticles, when they come in contact with the receptors or in the cell membrane, then what may happen is that they may lead to the activation of G protein. Now, this activation of G protein may lead to the activation of pathways which may increase the burst of calcium ions inside the cell. Then, additionally, what may happen is that they may lead to the production of reactive oxygen species inside the cell, as we discussed. So, this production of reactive oxygen species, this may be due to the peroxidases which are located on the membrane or which are present inside the cells, and then other ion channels may also be involved, and then another important key player is the NADPH oxidase. So because of the action of this carriers and the enzymes on the inside the cell, when they sense the signal from the nanoparticles, they lead to the production of reactive oxygen species. So this reactive oxygen species generation, they may further induce the signaling cascade, including the MAPK, means protein kinases pathways. So this phosphorylation of the protein kinases, now this may also be induced by the calcium bursts, which are caused by the activation of the G proteins. So the increase in calcium may also further cause ROS generation, or may induce the MAP, means mitogen activated protein kinase pathway. So because of the action of the reactive oxygen species or the induction of the MAPK cascade, now what may happen is that this MAPK cascade may induce the synthesis of different hormones involved in plant defense response. So the key hormones which are involved in the plant defense are the jasmonic acid, salicylic acid, and ethylene. So when these hormones are activated, these are involved in the normal response of plants to the pathogens or herbivores. So this will induce the synthesis of phytoalexins or compounds which help in response of plants to pathogens. Then additionally, what may happen is that these compounds may also induce the uh, activation of genes which are involved in the synthesis of secondary metabolite synthesis. So as we see in this diagram, so the production of the different hormonal hormones or the MAPK cascade or the reactive oxygen species inside the cell, they will induce the transcriptional reprogramming, or they will induce the synthesis of the secondary metabolite synthesis genes. So in addition, they will also produce the and genes activate the genes for the antioxidant enzymes. So these antioxidant enzymes, as we discussed, like the SOD, APX, or catalase, now the production of these antioxidant enzymes, they will be utilized by the plant cells to counter the production of ROS, or reactive oxygen species, inside the cell. So the production of reactive oxygen species as a result of the use of nanoparticles as elicitors will be countered by producing the antioxidant enzymes. So this is the antioxidant enzymes which are, am I audible now?
Yes, ma'am, you're
Yes,
audible.
madam.
You're
Yes.
audible from the
Yes,
very beginning. You started the session, you were audible from that time only.
no, actually, I see some interruptions in my
Ye-
network, so I'm just
Yeah,
confirming.
no, ma'am,
Yeah.
ma'am, you're audible now
Okay.
and visible also. Everything is now fine.
Okay, so what is happening here is, when the plant cells, when we use nanoparticles as elicitors, the nanoparticles enter inside the cells, they induce the activation of G proteins, and then once this is activated, they lead to the calcium burst or the production of reactive oxygen species through the involvement of NADPH oxidase or peroxidase or the ion channels. So this reactive oxygen species, these are produced inside the cells, which increases the oxidative stress inside the plant cells. So to counter this oxidative stress inside the plant cells, the antioxidant enzymes, their genes for the antioxidant enzymes are induced, and they produce the increase the production of antioxidant enzymes to counter the oxidative stress. Then additionally, another effect of all this is the induction of genes which are involved in the synthesis of secondary metabolites. So as the nanoparticles are perceived as elicitors, now they lead to the increased synthesis of secondary metabolites in the plants, and therefore what happens is that when we apply nanoparticles to the plants, they lead to the increase in the synthesis of these important metabolites or phytochemicals. So some of these phytochemicals, now see, they may help in fighting against the pathogens, means they are involved in plant defense. So therefore what happens is the plant, they grow better and they improve in development because of the increased production of these important metabolites. And in addition to that, we have higher production of the pharmacologically important compounds in the plants. So either in plants which are growing in the fields, that is in vivo, or in plant cells and tissue cultures, when we apply nanoparticles as elicitors,
Madam, you're not audible. I think again the network issue has started. Please bear with this. Let Madam join again and start the session. I think Madam has left. Please bear with us. She will join again soon.
I'm sorry again. So,
No, madam. Okay, fine.
so where did we missed?
Madam, you can show me the slides, then I can confirm. I don't
Okay,
remember the heading of the slide,
okay.
but I can
Yes,
tell you
yes, yes,
which
yes.
slide.
Sure, sure, sure. Okay, so did we
Uh,
receive
I,
conclusion?
I think it was the 14th slide.
14th, okay, 14th, okay.
After this,
So
you
this
started the
one.
15th. Yeah, after I think you yeah, no, the previous one, 14th one. And participants, please confirm. I think it was the 14th slide. Yeah, this one. Yes, ma'am.
Okay, so this one.
Yes,
So
ma'am.
did we? Okay, so you haven't seen this one.
No, ma'am. No, this was
Okay.
you were discussing this slide,
Hmm.
but precisely I don't remember which point you
Okay,
were
okay.
talking.
Okay, okay, okay. I will just summarize everything once again. Okay. So when the plants perceive the nanoparticles as elicitors, they activate the G protein, and then the pathway leads to the synthesis of calcium ions, bursts of calcium ions. So similarly, the uh, interaction of the nanoparticles with NADPH oxidase and the this leads to the activation of the peroxide pathway, which leads to the production of reactive oxygen species. So the production of all this protons, peroxide ions, and all this leads to the increase in the reactive oxygen species production or oxidative stress inside the cells. So this increase in the oxidative stress, they stimulate the MMTTC or protein kinase lead to this activation of genes involved in secondary metabolite synthesis. Another result is to counter the increase in the oxidative stress inside the cell, they lead to the induction of antioxidant enzymes, synthesis of antioxidant enzymes like the superoxide dismutase or SOD, then ascorbate peroxidase, and this one catalase, etc., to counter the oxidative stress inside the cell. So then another result of the activation of the MAPK cascade is the synthesis of the hormones which are involved in plant defense. So three main hormones, that is jasmonic acid, salicylic acid, and ethylene, they are involved in plant defense. So the induction of these hormones inside the plant cells, they lead to the synthesis of pathogenesis proteins and other proteins which are involved in plant defense. So here what we see is that the interaction of the nanoparticles with the plant cells, they are the main pathways they are affecting the cell is through the increase in the calcium ion concentration or the reactive oxygen species. So the increase in the reactive oxygen species or the oxidative stress leads to the production of antioxidant enzymes and the uh, secondary metabolites. So through this pathway, the secondary metabolites are genes involved in the secondary metabolites are activated. So therefore, what happens is when we apply nanoparticles as elicitors, the secondary metabolite production is increased in the plant cells. Now, these metabolites, they play different role in the plants. So in the intake plants, now they may increase the protection against the attack by pathogens, because the this one synthesis of the hormones, that is the jasmonic acid and salicylic acid pathways, are also induced. So therefore the plants has higher protection to diseases, and they also has improved growth and development. In addition, we can see that we can induce the increase in the synthesis of this important secondary metabolites which may have pharmacological or other industrial applications. So we can apply nanoparticles directly to the plants through the foliar or application or root application, or in plant tissue cultures to increase the production of secondary metabolites. Am I audible?
Yes, ma'am, you're audible.
Okay, so coming to the next one, that is toxicity and biosafety. So we have seen that we can induce the production of secondary metabolites by using the nanoparticles. But the application of these nanoparticles poses some toxicity and biosafety concerns, because as we have seen in the molecular mechanism, the elicitation mechanism involves the production of reactive oxygen species. So this reactive oxygen species increases the oxidative stress inside the plant cells. At certain levels, to counter this reactive oxygen species, we have just now seen that the plant cells, they respond to the this oxidative stress by producing the antioxidant enzymes. But if the oxidative stress is very high, then what may happen is that they may lead to oxidative damage. They may induce pathways like lipid peroxidation or DNA damage, which may be harmful to the cell. So therefore, we have to consider, and this we have to consider the size and concentration of the nanoparticles which are used for nanoelicitation, because depending upon the concentration, the toxicity may be higher. So therefore, whenever we use the nanoparticles, we may have to consider the toxicity or the toxic concentration, so that we can benefit from the induction or elicitation of secondary metabolites, but we will also have to see that they are not toxic to the plant cells. So this is another factor or concern. So in regard to this, now one important application nowadays is we can use uh, important surface coatings with eco-friendly agents like chitosan or citrate. So this is one factor. Then another one is, in addition to the toxicity to the plant cells, we will also have to consider the toxicity of the nanoparticles to human and the environment. So because this may have the chance of, like, the whatever nanoparticles that we are using, they may enter the food chain. So therefore, there we will it is very important to consider their toxicity. And when we use the nanoparticles in food crops, then their biosafety assessment remains one important aspect. So coming to the conclusion, just want to confirm once again, am I still
Yes,
audible?
yes, ma'am. Yes, yes, you're
So
audible.
this is, uh,
Now
thank
there
you
is,
so much.
I think again there everything is going smoothly.
So
No
this is the
problem.
last slide. This is the last slide. So thank you for bearing with me. So now, so coming to the conclusion. So now what we have seen is that we can use different types of nanoparticles for elicitation, that is enhancing the production of secondary metabolites in plants. So why this is important is we can use the nanoparticles to increase the synthesis of any compound, whether it may be a pharmacologically important compound, it may be a nutraceutical, or it may be any useful compound, or it may just be a compound which is important in plant defense, means the for the plant to protect itself from pests or pathogens. So whatever this defense compounds or the important metabolites, their synthesis in the plants can be precisely regulated by applying nanoparticles. So we have seen that we can apply the nanoparticles in different ways, either through the soil or by treating with the seed or by applying on the leaves of the plants. or we can, in case of plant cell and tissue cultures, we can add the nanoparticles in the medium. So we can use these nanoparticles to increase the synthesis of secondary metabolites. But the success depends on balancing the efficacy, means the its effectiveness, and we will have to see the biosafety and toxicity. So we have so from this we learn that we can use the nanoparticles to increase the synthesis of important metabolites in plant. So sometimes whatever metabolite that we are interested in, if we can increase the synthesis, then they can be helpful in extracting those compounds in large quantities, because most of the time most of these secondary metabolites are produced in very little quantities in the plants. Thank you.
Okay, thank you, ma'am. Except from those little technical glitch, I think the session was really interesting, very well explained. Now, I request the participants, you can directly ask questions to our madam. We already have two questions in the chat box. If they wish, they can also directly ask madam. Otherwise, I will read them later. If there is anyone who wants to ask question, can come forward. And I request you to, yeah, Annesha Chaudhary. Yes, Annesha.
Yes, madam. Yes, yes. I have one
Can you
query.
just
Uh,
tell me, yeah, excuse me, can you just tell me your affiliation before
yes,
asking
ma'am.
the
I am
question?
assistant professor in zoology from Lady Brabourne College.
Okay, now you can ask the question.
Okay, okay. Thank you, ma'am, for your very nice presentation. Except some technical glitch, it was very nice. There are different plant secondary metabolite active ingredients from which are used in different therapeutic purposes. I have a query whether the harassing nanoparticles can affect the therapeutic potentials of these active agents by chemical modification or by any other means.
So what you are asking is whether these nanoparticles can modify the bioactivity of the metabolites.
Yes, yes, ma'am. In their therapeutic, in terms of their therapeutic potentials.
That is totally another application. Totally another application means in
Hmm.
medical application. Yeah, this that is another application where, yes, if this compound, the compounds or the metabolites which are used as medicine, now their efficacy, now nowadays, that is nanoparticle delivery is one important aspect. So
Yes,
yes, that is yes, that is another important aspect, which is different one means that is in for medical purpose. Yes, the delivery or the bioaccessibility or the
yes.
bioavailability of certain compounds can be increased by treating with nanoparticles. That is one important aspect. Yes.
Okay. Got it. Thank you so much. Okay.
Thank you, Anisha.
Thank you, ma'am.
So
Thank you.
I will, yeah, if there is, I will give chance to other, but I have few questions in the chat box. I will read out to madam. The first
Yeah.
question is from Obhik Chaudhary. He asks that to study the effects of MPS in plant systems, are soil-based systems or hydroponic systems are better suited?
It depends on the stage of the study. Means uh, when we have to try the effects of the nanoparticles, now most often what we do is we start with in vitro. So we can we can start with in vitro, like we can study the pathway in vitro cultures like cell cultures or organ cultures or tissue cultures. So that is one pathway. And then another thing is we can after, like it depends on the, like for the efficacy, in vitro study is the initial study. Then in planta or in vivo, we can conduct either in hydroponic systems or in this one potted plants or in field conditions. So it depends on the stage of the study. So hydroponic system is also a very effective one, because in that you can you carry out the study under control conditions using only different hydroponic solutions where you can manipulate everything. But when you do the study under field condition, then you will have different other interacting factors or factors. So that will also that, but whatever nanoparticles that you want to try, ultimately it will, you will have to show its efficacy under field condition. So whether you try it in the first in the hydroponic or you try it in the potted plants or field condition is depends on the stage of your study or your choice. So hydroponics is also suitable because you can control a lot of factors or parameters in hydroponic condition, but in field condition, the ultimate effectiveness will be established when you try it under field conditions.
Thank you, ma'am. Thank you for your answer. Thank you.
Thank you, ma'am. Another question is from Oyon Dash. He asked, in terms of yield of secondary metabolites, how far nano elicitors are comparable with SA and JA spray or hair root culture?
Now, for this, there are many reports of the use of nanoparticles. Now, when we talk about the effectiveness, when we talk about the effectiveness compared to SA or JA or any other thing, now the thing is that one important aspect of using nano elicitors is that now when we use nano elicitors, as we have seen, now they have to be used in very small concentration or dose, because we have seen that when we use higher doses it induces toxicity. So compared to the conventional salicylic acid or jasmonic acids, then their effectiveness is great. Most of the time they are more effective at lower concentrations, because they because of their small size they can enter the cells easily, and they can elicit the, or they can induce the secondary metabolite synthesis pathway effectively. So in different studies in different plants, now most of the findings in the studies, what we commonly see is that now commonly the nanoparticles are more effective at lesser concentration. So this is something which is important, because which can be used for precision agriculture. So so we can apply these nanoparticles in very small doses and still we can reap the effect. So that is the importance, because most of the time what has been seen is that because of their higher penetration power now and higher this one, they can induce the synthesis effectively. So in when we compare it with the salicylic acid spray or the chitosan spray, or in vitro condition also, when we compare it with the general like chitosan, now the nanoparticles have been shown to be more effective at lower doses.
Okay, ma'am. Thank you so much.
Okay. Another question is from Arijit Das. He asked, are there any negative effects on the plants due to this nanoparticle exposure?
Yes, as we discussed in the toxicity. So as we saw in the molecular mechanism here. So as we saw in the molecular mechanism, now the molecular mechanism for the induction of secondary metabolites in response to nanoparticles one important pathway involves the production of this reactive oxygen species or the induction of oxidative stress. So what happens is when this oxidative stress is not very high, we are getting the benefit of increased synthesis of secondary metabolites. But to counter this oxidative stress, the plants, they are also synthesizing the antioxidant enzymes. So as long as the plants can counter this oxidative stress with the production of the antioxidant enzymes, now they may not be toxic to the plant cells, or the plant cell may be able to uh, this one bear the effect of the nanoparticles. But when we apply higher concentration of nanoparticles, when the oxidative stress is produced in excess, then what may happen is, as we have seen, they may lead to oxidative damage to the plant cell. So when the oxidative stress is high, they may cause lipid peroxidation, or they may cause DNA damage. So what happens is, mostly at higher concentrations, now mostly concentration is very important for nanoparticle application. So at higher concentration, they may cause excess reactive oxygen species to induce damage to the plant cells, and they may become toxic to the plant cells.
Arijit Das, your question has been answered, explained very nicely by Madam. I think all the questions were addressed very perfectly, answered perfectly, Madam. Thank you. Any more questions? You can ask directly. So I think there are no more questions. Your slide and your discussion was enough to make them understand without any questions. Yeah, ma'am, the discussion was very thoughtfully designed to offer valuable insights, and the discussion was very meaningful. It was exciting enough to engage the participants and give them fresh perspective on the topic. So, madam, thank you.
Thank
Thank
you
you for
for
joining
this.
us. Uh, Hopefully, we can meet again. Yes,
I thank you for giving me this opportunity, and then I hope that the participants found the content useful. So thank
yes,
you all
ma'am.
once again.
Yes, ma'am. Everything was very perfectly done. Thank you, ma'am.
Okay, thank you, thank you.
Thank you.
Then, uh,
Yes.
so can
You,
I leave
you,
now?
yes, ma'am. You can leave.
Okay, thank you, thank you,
Thank
thank
you.
you, everyone. Bye bye. Have a good day.
So we will wait for some time for Professor Shudip Kumar Ghosh. I think he has not yet joined. And hopefully he will join sharply at 3:30 p.m. and we will start our session, our next session. Okay, so please stay. We will we will start sharply at 3:30 p.m. I think Professor Shudhip Kumar Ghosh has joined. Let Sir confirm and we will start the session. Professor Shudhip Kumar Ghosh, are you joined, sir?
Yeah, Ajan, can you hear me?
Yes, sir, you're audible.
Okay, thank you.
Okay, so we are about to start our next session of this afternoon. So before we start, let me introduce Professor Shudhip Kumar Ghosh. He is a professor and former head of the Department Biotechnology, IIT Kharagpur, and a professor in charge of Advanced Laboratory for Plant Genetic Engineering, IIT Kharagpur. With a bachelor's degree in chemistry major and a master's degree in biochemistry, Dr. Ghosh joined the Department of Biochemistry Bose Institute as a PhD student in biochemistry. He completed his PhD work in plant biotechnology in 1995. After his doctoral studies, he did postdoctorate from Harvard University, Boston, USA, specializing in immunology and molecular and cellular parasitology. He was a visiting scientist at Harvard University and a visiting faculty member at Boston University School of Medicine in the Department of Molecular and Cellular Biology. He joined IIT Kharagpur as an assistant professor in 2000. He has been teaching immunology and immunotechnology to BTech and MTech students since 2001. Professor Ghosh has acquired theoretical knowledge, technical skill, and research experience in the field of cell and molecular biology, immunology, and biochemistry. He has about 130 publications in peer-reviewed journal with an H index of 48. He has served as sectional president of the new biology section at the 107th Indian Science Congress 2020. He has served as the chairman of the Model Curriculum Committee of the Biotechnology course for AICTE Government of India. Currently, he is also serving as a member of the Board of Studies of the Department of Microbiology of West Bengal State University and of the Department of Biotechnology of Haldia Institute of Technology. He is an elected fellow of West Bengal Academy of Science and Technology and a fellow of the Royal Society of Biology. With this, I welcome Sir once again and request him to start the presentation and address our participants. Thank you, sir.
Thank you very much for your kind introduction. So, before I start, I think it is let me tell you, like, it is difficult for the teachers to listen so many seminars or attending classes, I would say, as you have to appear for exam. So, at least give some test. Because we are, as a teacher, we are habituated to tell or taking classes. So, listening class is always difficult. I think your previous seminar was on plant, if I'm not wrong. And actually, what I will discuss in next 1.5 hour is what kind of research I am doing in my laboratory and some part of my own research during my postdoctoral. I will start with that and then switch to my own research. So, am I audible to all of you? I mean, hopefully.
Yes, sir. You
Yes,
are
sir.
clearly audible.
Okay. Let me share my slides first.
Yes, sir. Your presentation is also visible, sir.
Okay, so as I don't know whether it is given or not, I am talking about the encystation and cyst wall formation of entamoeba, particularly entamoeba histolytica. But I will tell you, I work with a model system because of some technical difficulties that I will come. And as a part or resident of West Bengal, I don't have to explain what is amoebiasis or what amoeba is. So you all are familiar with the amoebic dysentery or some other part. But amoeba, few information as an introduction I think I should give, which is the amoebiasis caused in human mostly by Entamoeba histolytica, which is the third most common cause of death in throughout the world due to parasite after malaria and schistosomiasis. About 100,000 people dies, mostly the children, and it is more common, as in tropical and subtropical countries where sanitary condition is not good. Actually, this particular disease transmitted through water or cyst contaminated food. So if our sanitary condition or the water is good, then it is very unlikely that we will have amoeba giardia kind of protozoal infection. And it is spread almost all over the world except North America and few part in Alaska and some part in Australia. Otherwise, it is common. Mostly it is traveled by the travelers because Mexico, India, Bangladesh, we all are carrier actually. Even we don't have the symptom or the manifestation of the disease, but all of us, those who are residing in this area, are amoeba positive. Fortunately, only 10% people get affected with this disease, and 90% we are the carrier. And a smart parasite, all of the parasite means it needs a host. So a smart parasite is the parasite which will reside inside the host but don't make any damage, because they maintain their life cycle within us. So if they make damage or cause disease, we'll take medicine and they will die. Okay, and their life cycle will be disturbed. So particularly what happened if for a good or smart parasite should be, for amoeba, there are two forms in their life cycle. One is the motile form, which which is there is no specific shape, and we are habituated or we are seeing this from our childhood that amoeba is it is say anybody, the good part of amoeba is anybody can draw it. Any shape you draw, you just make a nucleus in it and you can represent amoeba. But this amoeba is the motile form. Okay, just a minute. This amoeba is a motile form which can move, and this is called pseudopodia, by which they move and make phagocytotic cup, take their foods. But this form is not infectious. Infectious form is this. This is called cyst. The cyst is little smaller than the amoeba or the tropozoid form. Their difference is they have a very thick chitin wall, which make them resistant so that they can stay outside our body. The tropozoid, which resides inside our body, they cannot stay outside our body. So when they're coming out from our body, so they convert tropozoid to cyst, which we call encystation. So during encystation, a lot of things happen. The one very clear thing happened, from one nucleus they become four nucleus or four nuclei, and they make a very thick wall of chitin and different other proteins. I'll come into detail. So this chitin wall makes them resistant to heat, desiccation, pH change, outside harsh environment or drug entry. And this cyst can survive outside almost a month. If any water contaminated with this cyst we ingest, then this cyst through food, it cross the acidic region of the stomach and then go to small intestine. From after crossing this cyst germinate, we can say germinate or we call it excistation. So this excitation step, from one cyst, they make 4 of such trophozoite, which immediately divide into 8 trophozoite. One cyst is enough to cause infection. So how they survive? They eat our colonic bacteria when, and with the process they go, so they have nothing to do with anything else with our body. So the colonic bacteria, they just stay there. When they reach large intestine, what happens? There is no food. Their minerals get reduced. Food is not there, no glucose, no bacteria, and then they have water scarcity. So all these things give the signal to trophozoite that they cannot survive there and they have to go out. So that stress, like osmotic stress, food stress, give some signal which make them cyst. So my lab, which this research I actually started during my postdoc. So how this signal goes, how trophozoite understand that I have to do, what to do, and how they make this wall, that is actually I am trying to understand since last 30 years. So those who are interested to develop drug, they can develop drug, they can block the encystation. So if encystation can be blocked, cyst cannot be formed, so trophozoite will come out and they cannot infect us. So causing disease is, again, I am telling, this is not a smart choice of any parasite. Even, say, for example, if amoeba cause diarrhea, during diarrhea, this trophozoite will come out. They won't get time to make cyst. And trophozoite cannot survive outside maybe after a few hours, not even that. So those who are not familiar with amoeba, so this is the trophozoite cell which is loaded with vesicles or vacuoles. This is the nucleus. This is big, and these three are the cyst. You can see the size are different. They are non-motile. They don't move. Their metabolism is almost stopped. They don't do any metabolism. And these trophozoite, you see, this is how they move. Can you see the video? Is it moving?
Yes, sir, it was moving.
Okay, so this is just an example how we see amoeba under the microscope. These are cyst and this is the trophozoite. And if these are some still picture, what is we see? This is cyst again. You see this bright ring, white ring. This is we stained with calcofluor, where the cyst wall takes stains, which is bright blue fluorescence. 4 nucleus. This is the chromatoid body. Chromatoid body also a unique feature of cyst, entamoeba cyst. What is that? This is actually a collection of all the ribosomes, RNA, because they don't do any metabolism. Almost protein synthesis is not required at the last page of the encystation. So they just plaster together and save it. When they will exist, that means from cyst to trophozoite they will form. These ribosomes and all these things will come and reuse it. And this white part is a glycogen granule. This is again like a seed. They store the glycogen so that when they will exist, they won't have to worry for food or anything, so they store it. So these are 4 very specific features, and particularly when we check stool sample of amoebic patient, so chromatoid body is very clearly visible. We can see 4 nucleus. So this is the characteristic feature by which we diagnose the amoebiasis in stool sample. So what happened actually? In any unicellular parasite, bacteria, or any cell, even in our own cell, undergo different stresses, and which is common for amoeba. What are the stresses? Because amoeba resides in our colon, which is anaerobic. So outside the environment, they are having oxidative stress. Heat shock is also another stress. Glucose starvation, that means scarcity of food. This is a kind of stress. and nitrosative stress, which different kind of nitro compounds which give them. So as a single cell, they have to accommodate, or they have to change their character or pattern or protein or their target to save their. Normally what happened in later stage of amoebiasis, if anybody just affected with or infected with amoeba and not treated for a long time, they sometimes cross the mucosal barrier and goes to liver, and they cause liver abscess, which is not very at all a good one for any individual. So liver abscess, and you can imagine liver is full of oxygen. So they have a mechanism so that when they're residing inside the colon, they're anaerobic. When they are going to liver, they have a lot of oxygen pressure. So they have a mechanism how to handle this oxygen pressure. Okay, so similarly, they develop different kind of stresses, and food scarcity is also a specific stress which make them cyst. So what happened during encystation? I will show a few more videos. So these are the very early stage. Okay, normally when we culture amoeba, you see, so this amoeba, how they are moving. Okay, but as soon as we give them stress, like, I mean, I'm talking about in vitro culture, as soon as we give them stress, they become elongated. The same uneven shape amoeba become polaroid and they become elongated. And if you see, their movement is very much unidirectional. Okay, so they move unidirectionally. They don't move like randomly. Okay, so this is the first change you have noticed. So why this, or how this elongated cell and their unidirectional movement is helping? So you see, this is another culture condition when we put them little more in number and induced encystation by reducing the osmolarity, like salt concentration we reduced to 47% of the original medium, and remove the glucose. So it's a no glucose medium, so no carbon source. So what happened, if you allow them, this is a time-lapse video, if you allow them, so they move and ultimately they become aggregated. And I don't know how many of you have seen, if you can try, if not noticed before, if you see some ants are moving in a position, or if there are some food, lot of ants are there, you just put a drop of water, all the ants will be just gathered together and they make a cluster kind of thing. So any kind of trace, even we do, if there is suddenly some storm or something coming, all the people are coming and running in together in one place, okay, for safety. So this is a kind of, this clustering is very, very important for encystation. And in these clusters, there are some common or a community kind of signal they exchange to each other, which we are still looking for what molecule actually they are sending, how this, all the amoeba in a culture or in a particular place, they understand that they have to go which direction and they have to make cluster. This small molecule, it may be cyclic AMP, it may be something else. We are still looking for it. We tried different things, but something worked, some did not, and that is not satisfactory as much like so far we have received. So what happened? So first thing, they were amoeboid, then they become elongated, and they cluster together. So these three things are very, very important. If by any chance we can disrupt this aggregation, encystation efficiency reduced a lot. Okay, so, and another thing happening during encystation, when there are scarcity of food or anything, what happens? Cells are fused together, which is also happening in cancer cell and in macrophage. We call it giant cell. Okay, so as a first or very early eukaryotic cell, many properties what we see in our human are very similar with this. So if we consider the evolution, that evolution happened long back. We have a lot of modification. Our gene structure is not very similar, but the genes are similar. The protein sequences are different, but their functions are very similar. So I'm going to show you some more video what is happening. Just a minute. If you see this video, you see two cells. Okay, they fused. Here, two big cells, they fused, and you can see the surrounding amoeba, which is here. And this is when there are a lot of cells already fused this big, and the surrounding, you see the amoeba becomes smaller. So it's the scale is very big. You will see the two cells fused together. So this is in still. So what happened? This is the still picture of the previous video. These two big cells, we don't see very two individual cells, they are fusing at the early stage. We couldn't figure out, but when they become bigger, we can notice and record it. So this particular two big cells, they ultimately fused one. This is too big. You see the individual tropozoites are this big, and here individual tropozoites are small. Here even it's smaller. That means this cell size is very, very big. And when we stain the nucleus, you see only 4 nucleus here. This is little more. At the end, so many nucleus. I cannot count, which indicates these many cells at least fused together to make these big cells, and we call it multinucleated giant cells. And this is a very common thing happen in cancer cell also. How? Because cancer cell is also undergoing glucose test because they don't have food supply or oxygen supply that much, because their growth rate is very high. So we found some similarity what is happening in this single cell protozoan parasite as well as in cancer cell. After that, when these giant cells, these are the big cells, you can see this is a very big cells, and all these tropozoites are so small. Okay, this with respect to these big cells, this is the original size tropozoite. So if you take these giant cells to normal condition again, they started dividing. So this big cell divided into one, and then two, this again it will divide, and continuously it will keep on dividing. Okay, this will divide, and if you see the same thing again, continuously cell division is happening. Okay, we collected the giant cells and we see. So this is again a stress mechanism. So not that all cells, you see there are so many big cells, now these are comparatively smaller. I mean at the end, if you see there are so many big cells, but at the end of this slide or this video, you'll see most of the cells are become smaller or natural size tropozoite. So what I wanted to tell here is that if you put them in stress, first thing, they understand somehow, they elongated, they clustered. Within that cluster, some cells become bigger so that their total requirement will be less, and if they get a favorable condition, they become again a small tropozoite which will survive, or they become cyst. So what happened during encystation? So this is, I can skip. So you see this is again, so normally when cells are tropozoite, we make them video and track them with a software by ImageJ, which is freely available. You see these lines are the movement. You see these lines are very random. Okay, but when they become just polarized, their lines are very straight. So their movement are very straight. Okay, so this is a property of this polarization is must for or very much required for the encystation. If somehow this polarization and directional movement we can stop, they cannot aggregate and they cannot make cyst. And what is the reason behind it? This is already published in paper. You can see all this video in much more detail here. Due to time constraints, the video size and time is reduced. The cellular events of multinucleated giant cell formation and done by Deepak, one of my PhD student. He passed away 3-4 years back. So all these things happening, all important event means this is the actin. Actin is the structural or scaffold protein. So during normal condition, actin is localized like this, and gradually you can see this actin is changing their location. Ultimately, when they become Polaroid, all actin come from the posterior region. This is the front side. So they will move from this side to this side, and all actin come here. So actin polymerization, depolymerization, and their reorganization during this movement is very, very important. Okay, that is also we figured out. And these are the, this is another picture, set of pictures. This is how you will see actin when you localize them. What we do actually, this is very not very tricky. There is one reagent called phalloidin, which is a plant alkaloid. The phalloidin labeled with a fluorescence tag is commercially available. You have to fix the cell and add that reagent. So wherever polymerized actin is there, they go and bind, and only then it is fluorescent. So at normal tropozoite stage, what we see is all these rings are the phagocytotic cup. Okay, so they are always ready to eat something. Okay, it is actually so this is like a cup. So suppose this is the amoeba, and actin is just below this membrane. Okay, so this cup, and if you see from the top, it will look like this. So this black part we see as a ring. So whatever this ring or this ring, this is a phagocytotic cup, and here it is even clear. This is another cup, this is another cup. So with time you can see the number of phagocytotic cup is decreasing, and actin localization is also decreasing. So they become in the Polaroid position, and they come in the front, and gradually they come in this region. So they become Polaroid, and this is also, it's a lot of work. This is a summary of a big this is also published in Infection and Immunity. So those who are interested to learn in detail, this is almost 25 pages article, and we are fortunate that this figure that which I saw, which become in the cover page. So details, I'm not going in detail because little more thing I have to discuss, I thought. So again, I'm coming stress. So now what is happening? So how or what kind of protein or signaling system is there in amoeba, so that they can understand that some changes happen, like food is not there, or the heat is there, or oxidative stress is there. So to understand what exactly the beginning part before this motility change or the direction change, what happening. So I'm not going again detail. There are few. One MAPK kinase, one AMP kinase. AMP kinase is the kinase that acts when AMP is increased. When AMP of a particular cell will increase, because the ATP production is less, because metabolism is less, no food. So ATP will break down and more AMP will come, and this AMP, higher AMP concentration will activate a kinase, which is again a stress response, like which is also very much controlling our glucose homeostasis, like insulin secretion, insulin function. One protein kinase and one stress responsive topoisomerase we already cloned, explained, and published. EI MAP kinase, this EI stands for, actually I forgot to tell, EI is Entamoeba invadens, which is a reptile parasite very similar to Entamoeba histolytica, but they cause disease to reptile. Why we are using this? Because Entamoeba histolytica we cannot make cyst inside the lab. So we have to depend on human system only, which is then it is working is not possible. All these time points and things are not possible. So we work on entamoeba invasion system. Not only we, whoever is working on encystation of amoeba, they are working with entamoeba invadance. So MAP kinase, which I saw or tell you, MAP kinase is very important because when we knock down the MAP kinase, that means we reduce the expression of MAP kinase. What we see, the aggregation become less. Aggregation, you see this is in the control aggregation, and this is the aggregation when the MAP kinase we reduced by molecular silencing, gene silencing. And when a gene silence, I mean this percentage of encystation efficiency is also reduced to 50%. It's not zero because we cannot completely abolish the MAP kinase, we just reduce certain percentage. So, and when you see the chitin formation, that encystation or cyst formation, this blue color is indicating the chitin outside the cyst. This is cyst. You see in a control cell, this is very much uniform, like woolen ball, but here it is just very random or not properly make the wall. And this is a scanning electron micrograph of the control and MAP kinase silent cell. You see the chitins are not very much okay. I will come again with this, how this thing is happening, that is why. So now coming to the wall formation story, this is actually an aggregation of 12 hours. And if you see, these are the tropozoite outside this aggregation. When we stain the cyst with fluorescence dye, that dye is chalcofluor white, which binds almost all the carbohydrates like cellulose, chitin. So you see all these white dots are the cyst. And if you see carefully, because these two pictures we superimposed, you see cysts are mostly within this aggregate. Wherever aggregates are there, cysts form there. So all these free tropozoites, very few cysts or no cysts. With time, like after 48 hours, the aggregation is still there. Many of the cell inside this aggregation become cyst. You see all the white spot of cyst, and this is just enlarged picture how the cyst look like. This very circular and uniform chitin wall is there. This white ring is actually due to the chitin presence in cyst. By the way, if you have any question in between or anything is not clear, please let me know. Okay, so you can interact me, you can stop me, that's fine with me.
Okay, sir. Okay, so you all have listened. Sir is interested to take questions between the session also.
Yeah,
Actually, we were planning to have the interaction session at the end of your discussion.
Yeah, but at the end of the discussion, what happened? I have to go back again on.
Okay, sir. Okay, sir.
If some part is not clear, then it is not good.
Yes, sir.
To follow the rest of the part will be not interesting actually. Those.
Okay.
Yeah, hello, sir. This is Prijit Nambiar from Mumbai. So I was just wondering, like, whether this multicellular fusion that takes place, does it take place normally in the colon of a human, and does it has any role in pathogenicity?
No, this is first thing is whether it is happening in colon or not. It is not known because no stool picture we have seen this so far. Number one, the event is not huge in number. What you saw, this is after culturing lot of cells and collecting them, making them concentrate to take picture. Number three, pathogenicity means they can't do anything until unless they become the tropozoite again. So if they can survive in the stress condition and make the giant cell and come back to tropozoite again, they can again infect. That is the only relation with pathogenicity, not directly cause any problem. Because what happened normally, if amoeba, say I said already that in India or particularly this eastern region, tropical part, most of us are having amoeba inside our colon along with the gut microbiome. But they are eating bacteria. They're going to large intestine, make cyst, going out with the feces, contaminating the environment. And if any of us taking this contaminated food or water, we will be infected again. So there is no symptom, nothing happened. But what happens sometimes, for unknown reason, they start invading the mucus. So when they start invading mucus and enter into the bloodstream or in liver, so that mucus invasion caused the dysentery. So that's how the mucus came out with feces or stool. That create problem. I mean, if it is serious, then it is caused blood dysentery. But otherwise, they don't do anything. They don't do any pathology. They stay peacefully. They use our internal gut microbiota for food and spread. This giant cell directly not involved in any disease causing or pathogenicity. But if they survive, they will do it. So that way it is related. Is that clear?
Yes, sir. Yes.
Okay. So if you see these two pictures, this is a tropozoite. If you see, this is a transmission electron micrograph where you can see the membrane of this plasma membrane. And here, this is the cross section of cyst. So above this plasma membrane, you can see this is a very thick wall. This thick wall actually chitin wall, which is present in cyst. So that is totally absent. So what during, and it was known, I mean, this presence of chitin wall cyst is known from long time. But when I started my postdoc, so what we started with, the question is how this chitin wall is formed. Okay, so first, this one was answered long after I joined at IIT Kharagpur. We figured out the chitin synthetic pathway or all the genes present and what is happening during, and this is the last step. It started with glucose again. So normally when there is tropozoite, they need energy. This is anaerobic fermentation. So glucose goes to glucose 6-phosphate and then fructose d-fructose 6-phosphate and then go to glycolysis for energy production. But during encystation, the same fructose 6-phosphate enter into another pathway, which ultimately by chitin synthesis make chitin. So there is a competition between which way it will go, whether it will make chitin or make energy. So these are the gene ID, and we saw most of the chitin biosynthetic gene are expressed during 9 to 12 hours maximum. This is the reverse transcriptase PCR from mRNA. We see that brighter band is more expression. So they express mostly between 9 to 12 hours. Okay. So after that, what we see, we again downregulate some gene and ultimately figure out that when there is no glucose, because we said that scarcity is there, so from where the glucose is coming. During tropozoite stage, extra glucose converted into glycogen. But when the food scarcity or glucose is missing or no glucose is available, then stored glycogen break down to glucose, and that makes chitin and the required energy, because they also need energy to make protein and all other things. And we verified the expression of these genes. This is published. And then the total composition, what we found in cyst wall, not only chitin, there are some chitosan is also there. Those who are not familiar with the chitin, chitin is a polymer of N-acetyl glucosamine. Okay, N-acetyl glucosamine is a monomer, and this is 1,6 linkage. They make chitin, and when this N-acetyl glucosamine is deacetylated, we call it chitosan. You might have heard about a lot of, or we are going to listen a lot of nanoparticle or nanotechnology seminar where the chitosan nanoparticle, chitin nanoparticle are very common, and chitosan and chitin are very common and abundant polymer. Actually, this is the second abundant polymer after cellulose. Okay, a lot of all the sea animals, or many sea animals, I should not say all, have chitin. So chitin source is very cheap. So people are making chitin nanoparticles. So chitin, chitosan both are there present in the cyst wall. So what we did, let me check what is the next slide. No. What we did, we isolate the cyst wall. We just we sonicate them and collected this chitin wall. Okay. And this chitin wall, we then treated with SDS. After SDS treatment, we run a 2D gel, two-dimensional protein gel electrophoresis. And from that, we spotted that time when I started this working here, you can see the publications at 2000, 97, 1999. So it is long back because the genome sequence published, amoeba genome published in 2005. That time genome sequence was not known, gene was not known. So what we did, we ran a 2D gel and individual spot we cut it and we compared with the tropozoite and the cyst protein. Whichever is different spot, we cut them, we did the mass spectroscopy and protein sequencing, and ultimately we figure out what gene is there. Then with a standard protocol like cDNA library screening, genomic library screening, we identified chitinase was already known by that time. Chitinase is the enzyme which breaks chitin. Chitin synthase, definitely it was there, but gene was not known. We identified Jacob is one structural protein and JC another structural protein, all published long back. So what is happening? So what we figure out that at the 12 hour, this is a immunolocalization. If any of you are not familiar with immunolocalization, just let me take a minute. If any of you are just not familiar with immunolocalization, what happened in immunolocalization? So suppose this is the amoeba. And I would like to know where any particular protein, so everywhere protein is there, right? So if I would like to know where the protein of my interest is, so similarly one protein is say Jacob. So what we did, we ultimately identify and then clone, then express, sounds very simple, but it's not. Express, then purify the protein and raise antibody against this Jacob. So what is the immunostaining? So first we take the cell, we make it permeabilize with detergent and add this antibody. Okay, so this antibody will go and bind wherever the protein. Suppose this is the protein, so antibody will bind here. We cannot see that. Then we add another antibody which will bind to this antibody, and this antibody is labeled with a fluorescence probe that may be red or green depending on whatever system you have or you want to know. And you can buy red, green, yellow, blue, all are commercially available. This is we call secondary antibody. This one we call secondary antibody. So what is happening? We take the cell, we first incubate with the primary antibody. This primary antibody will go and bind, then we add the secondary antibody, and this secondary antibody, let me change that. Okay, this secondary antibody is very hard to see, will bind here of the primary antibody, and that is labeled with fluorescence tag. So now if you see under the microscope, you can see the color or the light. That's what we did here. So we use anti-chitinase antibody labeled with a green fluorescent secondary antibody. So wherever chitinase is there, it is going to bind. So you see chitinase is present in small dot all over the cell. And with time, 12 hours, 24 hours, 36 hours, the amount of chitinase is increasing. You see more amount of protein means more amount of fluorescence, and again diminishing. And if you see very carefully, there is a nice ring So it was inside completely, and then it become or localized in a very nice ring. Similarly, the Jacob we localized in 12 hours, it was inside the cell, and within 24 hours, it is on the membrane. And 36 hours, it is inside as well as in the membrane, and 72 hours, it's mostly in the membrane. JC, at that time, what we couldn't figure out because of the technical problem, we corrected it very after almost 10-15 years. So JC, initial level there was no expression. It started at 36 hours and eventually it goes to membrane. So 3 different protein, 3 different antibody, and the secondary antibody, one is green, another red, another blue. And then we superimpose this. Ultimately what we see, green and red is not together. They are different, but eventually they all make the membrane. Okay, so what actually happened? This is the chitin fibril like this. Okay, and these proteins are like this. So if I go back, I can tell you the mechanism a little bit. So recently that same Deepak Krishnan who figured out that this chitin wall formation, they started from a single point. So this blue is the chitin formation. So this is the cyst. In cysting cell, I should not say cyst, chitin formation starts from a single point and they gradually cover the whole thing. And this is a picture of multiple cell, and 48 hours, the whole cell is blue. That means the chitin covered it. So he took some nice picture. So it started in one point and gradually it is increasing and ultimately covered the whole cell. This is a single slice and this is the whole cell 3D. So they took a different slice of the cell. This is through confocal. So different slice of the cell. So what is happening? So if this is the, if this is the cell, if you cut a section, you will see like this ring. But if you take picture of different slice, many pictures, and put them together, make a G stack, we call it Z stack or G stack, then this kind of complete picture will come. Okay, so this is one observation that was not figured out before. And eventually what we found, this is very interesting to me. I don't know how much interest we will find. So initially, if you see this picture, there are a lot of phagocytotic cup. Okay. There are a lot of phagocytotic cup. But if you put them during encystation, all this actin, which was took different shape during this motility, they become peripheral. All the actin become and make a cortical ring. You can see it better. Okay, so this cortical ring formation is essential. Why I am telling you? So if you see this picture, it's not here. If you see this picture, sorry, if you see this picture, I don't know whether how good it visible to you. You can see the actin ring formation. Can you see all of you can see the ring?
Yes, sir.
Okay, so in that ring, same cell, when we see the chitin synthase, we localize same way by immunolocalization. Chitin synthase at the very early stage, it was inside, and then it come in the membrane. Okay, so chitin synthase deposited on top of actin. And wherever chitin synthase deposited, the chitin synthesis starts. This blue is the chitin synthase. So wherever chitin synthase, the green is there, only that place the chitin synthesis is happening. So that was very interesting finding to us, because this picture it's very easy to tell in the seminar, but it took months to get this picture. And I'm fortunate that I had Deepak as a student, because very few students in my last 25 years of research, individual research career, I have found those who are interested to see it under the microscope and constantly in dark room spending so much time. So wherever the green, that means wherever the actin ring is there, the green is there, and green means chitin synthase, actin is forming. Okay, so this tells us that this actin ring formation is very important. And we also prove that when we block the actin polymerization by different drug, like there are few drug available which can block the actin polymerization, then chitin synthesis completely disrupted. Okay, so this is, I try to make a three-dimensional picture. So this is the chitin, actin ring, the red one, and you see the chitin synthesis is increasing, and then one nucleus become 4 nucleus, and all the actin which was in the form of ring, they just break, and ultimately inside the cyst all actin disappears. So we'll have a very nice chitin ring and 4 nucleus. So this is the mature cyst. Okay, and the first proposed model was published, that was Wattle and Dwab model. Wattle and Dwab means this is what is happening. Now let me tell you in brief what biology is happening. So first, this is the cell. First, what happened? Jacob protein are inside the vesicles. Okay, you may not remember, but Jacob was all big vesicles. So first thing happening, actin ring is forming just in the cortical region of amoeba from inside. Then Jacob go and sit in membrane. Okay, like this. I'm not complaining. Jacob has chitin binding domain. Jacob is a lectin which has a chitin binding domain. Then inside, chitin is also formed. Chitin goes there, or chitin synthase goes there, and chitin fibril are making this. So what is happening? So Jacob is like this, which are these sticks are the Jacob, and chitin fibrils are making like this. So this Jacob and chitin is making this structure, which are very familiar to you. All the hut or mud house is like that, which is normally known as wattle. And what we do after making wattle, we put some mud on top of it to make it waterproof. On top of that, there is a third protein, JC, which synthesized later, which is that mud kind of thing. So they cover this whole structure and make the wall. And when JC come, it become waterproof or water resistant. So that is the model, and this paper actually discuss all in much more detail. Many more figures are there. So this figure, I think it is little detail is. This is evidence for Wattle and Dwab model of the cyst wall of entamoeba, which is published in PLOS Pathogen. Okay, so and that gives this model, and that still today, or till today, it is the only acceptable model of entamoeba and cyst wall formation. So anybody working, and this published in 2009, if I am not wrong.
Sir, sorry to interrupt. There is a question.
Yeah.
Question in the, since you told that you want to entertain the question,
sure,
so
no
I
problem.
just interrupted.
No problem.
Manashsi Mandal has asked, why does entamoeba histolytica not insist in human tissue?
Just a minute. Let me just finish this slide.
Okay, sir.
I will answer. So So this 2009 paper was the only model, and then in 2024, last year, we published another model, the novel inside of wattle and daub model, the role of actin cytoskeleton and chitin synthase. So just to add actin and chitin synthase, what they are doing during this process, it took almost like 14-15 years. So you can imagine a little bit. It's not the only thing I am doing. So it is time consuming. Okay. So both the paper are giving all the detail due to time constraints, and all the experimental details also I don't want to discuss here, which will not be interesting. So now question is, why Entamoeba histolytica is not making cyst in human tissue? They are not making. First thing is, amoeba, they don't make cyst on tissue. They are outside. They don't invade. They are not intracellular parasite. They are external parasite or extracellular parasite. Second, I did not tell that they did not make cyst, because inside our body they make cyst. But inside our body, if they make cyst, we cannot work with that, because we have no control. We have to do all this study culturing them inside the lab. We can culture Entamoeba histolytica, we can culture Entamoeba invarians, but outside the body they don't make cyst. If you ask why, I don't know, and it is not known to anybody in the world. Nobody knows. If I know why, then I will solve and I will make it cyst. There's only one paper who reported that Entamoeba histolytica encystation, but I tried a lot and many other people tried, and that was not reproducible. They are going up to certain level. After that, they don't make the complete or mature cyst. If you take mature cyst, if you boil with SDS, nothing will happen. They are so much resistant. But histolytica we cannot make. Is that
Okay, sir.
Okay, so sir, there is another question from Manasi. Does chitin dissolve during excistation?
No,
And no.
it is not known, but they break. They break and the tropozoite come out like the germination of plant seed, like the root. They come out first, or the hypocotyle, they come out. That way they come out. Not they don't break or completely dissolve. But ultimately they eat it, because they are very good eater. They can phagocytosis a lot. So if you exist them in vitro, or they take the cyst and put them in the regular culture, after certain time, like 12 hours to 24 hours, you will see a lot of chitin cell are there where nothing is inside. But if you incubate them for longer, you won't see anything. That means they eat them, they digest them.
Okay, sir. Another question.
Yeah.
Another question from Prajit. How does the time-dependent synthesis of different molecules occur? This is a question, and another is actin followed by chitin, and then chitin proteins. This is actually the whole question he wants to ask.
Yeah, what I want to ask is like the chitin, actin is formed, then the chitin, then the lectin proteins come. Like there is a time dependent synthesis,
No,
right?
it is not.
So what?
Actin is not come. Actin is always there. You see the normal, there are so many phagocytotic cup. As soon as you put them in encystation medium, that means no food, they don't have anything to eat or drink, right? So phagocytosis, pinocytosis, they don't need anymore. And the actin, the uneven cell become polaroid. So actin went to the posterior section. Then they become circularized. And during circularization, they give the actin. Actin actually what it is doing, it is not taking part directly. You know, they are making the roof. When you are making a concrete house, when you are making roof, what you are doing, or what the mansions are doing, they are giving a lot of support by bamboo and then make a flat surface on top. Am I right? On top of that, they put the concrete like stone, cement, sand, all the mixture. So if you just put them cement, sand, suddenly in a blank space, what will happen? We cannot make roof. So you need a support, a scaffold. So actin actually basically giving the scaffold kind of structure. On top of that, chitin synthase go and sit on top of that and make the chitin. And when the roof is done or solidified, what we do? We take out that scaffold and all the bamboo and the wooden support from the bottom. Similarly, we don't need the actin anymore. That actin again depolymerize, because actin polymerization and depolymerization is the regular feature of all kind of cells. The cell division, cell movement, cell migration, actin is always protein trafficking, vesicular trafficking. Actin is not just a protein which will make and sit there. This is a monomer. So globular actin will polymerize, make a chain, do something, then when the job is over, they again depolymerize, again that monomer will polymerize somewhere else for some other purpose. Here also similar thing is happening. And what was the question of the time dependent one?
Yeah, like how is it time dependent? Like, so after chitin is formed, then the lectin proteins are formed. Like, are they
that time dependence is everywhere. Like when I'm just giving example, you may understand. This is a gene regulation way, how gene is regulated. So when you eat something, or say a breakfast or lunch, what will happen? Your blood glucose level will increase. That will give the signal to your pancreas beta cells, which will produce insulin. That insulin will go and bind to liver and muscle. They will produce the enzyme which can make glucose to glycogen. That enzyme will secrete and make the glucose and glycogen. As soon as the glucose level decrease, insulin secretion will decrease. When it goes down even below, then the same signal will go and tell the another cell of pancreas to make another hormone, right? That what it will do? It will tell the same glucose liver cell and muscle cell to produce enzyme which can break glycogen so that they can produce glucose to maintain the blood glucose. So these regulation, spatial and temporal regulation of gene expression is everywhere. Here we don't have any spatial because it is a single cell. We don't have liver, muscle, heart, kidney kind of thing. And in amoeba I'm talking about. Okay, so this is actually all caused by signal transduction pathway. And what are those protein? MAP kinase is one, protein kinase we have this, AMP kinase is one. Okay, so they are giving the signal to particular transcription factors, and that actually controls what to do and what not to do. Okay, and that's how time dependent is the temporal control and tissue dependent is the spatial control, right?
Yes,
Insulin.
sir. Another question has come up, sir. If you are done with the answer, then
Yeah, I'm done.
It
it depends who asked the question, whether it is clear to him or not.
Yes.
Okay.
Okay, sir. So another question has been asked by Nobonita Ghosh. What is the effect of acidic pH of stomach on the cyst? How do they exist inside a human host?
Acidic pH is not doing anything to cyst, because cyst they don't allow the acid to come. But it may be possible that this acid exposure can make this cyst a little fragile, so that when they will reach to small intestine, their germination or coming out will be maybe little easier. But in vitro, that means in culture, we don't need any acid treatment. If you just take the cyst, wash it nicely, put them in regular medium, within 12 to 16 hours, they will start exist. But acid can just make them little fragile, or the hydrolysis kind of thing, some molecule, some protein maybe hydrolysis, but they cannot make any damage to cells. They have to, that is, if there is anything, it is beneficiary, not in the other way. May I proceed?
Yes, sir. Please. No more question up till
Okay.
now.
Okay. So then we started thinking like if there is no food, if they're making cyst, there's so many things happening. By now you can, I hope you will little bit you understand. So many proteins are synthesizing, so many transport, these, that. So energy is required, food is not there. So what is happening? How they are getting the food? So one idea came, one of my students, and we talked together and figure out like that cluster or the aggregation where they aggregate with a large number of cells, lot of different things are happening. This kind of aggregation is happening in bacteria also. They make biofilm, so and they give signal to each other and to do what not to do kind of thing. So here we thought, and which was not very like effectively published, or but there are some indication in amoeba that autophagy may be there. Autophagy is happening. One Japanese scientist, Tomoyasu Nozaki, he is working in a similar field. So he published a paper, and in one place he saw that one autophagy gene, ATG8, he saw that. So maybe there is some autophagy. So autophagy is there means cell will try to survive for longer time. And at the same time, some cell will die, lot of cell will die. And if the cell die, what is happening? How they are dying, and are this or the dead cells are the source of food to other? If it is bacteria, then it's okay because in the stationary phase it is regular feature. But in eukaryotic cell, it cannot happen that way. So if dead cells are the source of food for the other survived cells, then something else is also necessary, which is apoptosis. So we started together looking for like whether these things are happening or not. I'm not going to go in detail here much. So what is happening? We identified one apoptosis inducing factor, AIF. Normally what happens? Apoptosis is done in higher eukaryotes like in human or other higher eukaryotes. There is a specific gene or effect of a gene called caspase, which is a cysteine protease. That cysteine protease is that kind of caspase kind of cysteine protease is not present in amoeba. Amoeba has 8 or 9 cysteine protease, but they are not causing apoptosis. Another caspase independent apoptotic pathway was reported in human, particularly in cancer cell, called AIF or apoptosis inducing factor. We identified that, and we saw that this apoptotic morphology is happening when there is a drug or oxidative stress. All of you know that how to treat amoeba giardia is the metronidazole, tinidazole, like Flagyl or O2, Norflox TZ, or everybody is doctor in our surrounding. If there is any diarrhea, anybody will tell, okay, take a Cifran CT or Norflox TZ. That TZ or CT is the tinidazole, O2 is the ornidazole, and Flagyl is the, or Metrogyl is the metronidazole. All three are equally okay. Only their side chains are different. They work. So if you give them metronidazole in culture, they undergo autophagy, and if you add too much drug, they will die. If you just sublethal dose, after autophagy, they undergo encystation. So even the drug also can induce encystation. That means if amoeba, particularly I'm talking, and it may be partially true for giardia also, they are very similar in nature. Amoeba, giardia, trichomonas, all three protozoan parasites are very similar in many properties, not in the disease point of view. So this drug treatment is also a stress, and that also cause encystation. Okay. So when we are looking for this, so let me skip that. So a putative apoptotic inducing factor was predicted, we characterized, and we saw that when the amoeba, normally it is there. So if you induce that, they release, and ultimately chromosome degradation, all apoptotic DNA fragmentation, everything is, it's a bifunctional protein. It is a mitochondrial protein, but amoeba and giardia, they don't have a regular mitochondria, though are unique. They don't have mitochondria. They have mitochondria like vesicles, but not at all. They don't produce energy. They don't have DNA inside it. So we don't know. We are still looking for it, whether the AIF from where it is coming, but they induce apoptosis. And this apoptosis inducing factor like protein mediated stress and metronidazole responsive program cell death pathway in amoeba. This is again we published in 2023. It's molecular microbiology. All are available. Sorry, all are available. I am not going detail. And what is happening in amoeba histolytica? If you give them stress, particularly oxidative stress and drug, AIF increase, and they started dying. So what is happening? Our idea or my idea is like that aggregation, make them together. Many of these cells, due to scarcity of food, are dying. And they are not just dying like anything. They are dying by apoptosis. And when apoptosis happens, it is different from rotting of the cell or the necrosis. So apoptosis happens, this sink becomes smaller, DNA degraded, and that small cells, they eat. So those cells who survive, they eat the dead cells. One thing we have to remember for unicellular organism, it's not like higher eukaryotes. Like if suppose we started with 1 million cells, we give them treatment. It is true for bacteria also. We give them treatment or some stress, no food. Every cells die but one. So if one of them survive out of million or billion, that one can make million or billion in few days. Okay, so for them it is not that they're lost. And we have to remember another point, what after studying parasitology research, before that I did not have any biology background, chemistry, biochemistry, none of them. I have never read geology, botany as such. But what I realized that human came many years later than these organisms came, like all the bacteria which are causing disease. So when we evolved, we became host to them. Amoeba is surviving, as a topic of today's, or any other bacteria, surviving much or many more million years before than us. So when human did not exist. So they know how to tackle the environment, they know how to tackle the situation and can survive. So still they are maintaining and we are fighting. And because of the evolution, we became host for them. They were not the pathogen for us. The unfortunate part is this. So they have all the basic mechanism how to handle stress and the adverse situation. And this is one. So whenever there is an adverse situation, they cluster together. Some of them die. Some of them survive on the dead cells and waiting, and they try till end or the last cell. By then, if environment become favorable, that single cell become million again. And that's how they maintain their population. So this is one of the important things. And the next, and this is autophagy is cross-related, and this is done by Annesha Bandyopadhyay. All of them are already become doctor and doing postdoc now. This is a very recent work. So those who are working, how this time is maintained or the sequence is maintained. So what we found one transcription factor, tail homeodomain. Transcription factor tail is tri-amino acid loop extension. This tri-amino acid loop extension is the name. It's a homeodomain transcription factor which regulates many things in all over the animal and plant kingdom. So it's not there. So what we saw, that these transcription factors are present, we named it EIH box 1, present in the cytoplasm. But if you give them stress, you see when you give them serum starvation or glucose starvation, almost all enter into nucleus. Because transcription factor, what is their role? Transcription factor goes and binds to the promoter sequence and transcribe different proteins. So these tail homeobox protein we identified, which is encystation specific. I did not put that here. And like they increase up to after 6 hours to 12 hours. So this is the initial signal. And we find their binding site also. They bind in the octamer or in the hexamer, TGA, CAG, and TGAT, TGAT. So whichever promoter of any gene having either these or together, they will be controlled by this. As a result, we figure out, we did the RNA sequencing analysis. So first we silenced this tail homeobox protein. So we have a control, which is everything normal. And we have silenced. Silence means this gene is not expressing properly. And we did the RNA sequence expression analysis, and we figure out some genes are overexpressed, these genes are upregulated. I cannot read which is this, and you cannot too. You don't need it. Only thing is that some genes are upregulated and some genes are downregulated. If you knock out these genes or knock down these genes, that means these transcription factor regulates some protein expression either way, either increasing or decreasing. Very common thing is that this protein in general, what you see, all the metabolic gene is downregulated because we don't need it, and some other genes are upregulated. And what we found that if you silence them, chitin synthase, Jacob, these are downregulated, but JC is upregulated if you downregulate it. And interestingly, many proteins which are involved in actin polymerization, Rho gap, Rho A, gelsolin, vilin, and PI3 kinase. So these proteins are upregulated. This blue is the silence and control, and this is, so these genes are upregulated. Here you will see downregulated, but why downregulated? Because we silence the gene. So if you don't silence the gene, they are helping the actin polymerization, and as a result, we see that, I don't know, you can see this picture or not, even after putting them in starvation, they don't make polarized. They are random, so they cannot move. They cannot, their aggregation is less, their eating capacity, like this is the phagocytosis. You see part cell, so many bacteria is inside, but when you silence, number of bacteria is many less. So all the actin dependent pathway are blocked or reduced. And what we saw, that actin ring formation during encystation is also hampered. So as a result, in silenced cells, in control cells, chitin wall formation is so nice, but in silence, chitin wall formation is disturbed because it is disturbing the actin. So this is one of the gene who was asking of the controlling. This is one of the transcription factors which is controlling actin polymerization control as well as other gene expression control. So if we downregulate, amoeba cannot make cyst. Okay. Another, so now what happened? Let me go. This is, we also published in Molecular Microbiology. This is Sneha Pandey, Meenakshi Pandey. This is another Meenakshi's paper, 2 paper tail homeobox protein, how it is controlling the actin dynamics and cellular activities in entamoeba invadence. Then the last paper, which is published in last 10th November. So this is, let me read the title, the G alpha subunit. I don't know whether you are aware of G alpha subunit or not. There is a GPCR concept is there, which is a receptor, G protein coupled receptor, where G protein actually have 3 subunits at the inside the cell, which is alpha, beta, and gamma. Amoeba has only 1 alpha, 1 G alpha subunit, 2 beta, and 1 gamma. So she worked, or her PhD work only on G alpha subunit. He again silenced and saw the effect. What we saw is there is, can you read the inside? What is written? Gene name?
Yes, yes.
Okay, so because this is too small. So this is the GPCR. So what actually, summarizing the whole thing, what I said, this is the GPCR present in the membrane of amoeba. So any kind of external stimuli like serum factor, starvation, all this, some information came in the yellow, this is the ligand part, that gives the signal. So when that signal binds from outside, this GPCR activate the G alpha. This G alpha activate the chitin actin polymerization thing. By this, they also activate this EIH box, that means the transcription factors. These transcription factor goes and bind to the DNA and transcribe the different genes. What are those genes? They are the JC, Jacob, chitin synthase, chitinase, chitin deacetylase, and so that these proteins which are not present in trophozoite, all these genes which are making cis like chitin synthase, chitinase, JC, Jacob, none of them are present in the trophozoite form. So when they get signal, only then they started synthesizing, and when they are synthesizing, they are doing their job because they know what to do. So that is now up to we are. So we started with just what is there in the chitin wall to now we identified G alpha, one of the student. So all this name written in yellow are involved in this NC station wall only. All of them up to Silpa Sarkar all are awarded. Shubham is working on the G beta and gamma subunit. Amrita is working on this topoisomerase. Tiasa is working on chitin synthase. Susanto is working on helicase, because one of the important thing which I did not cover here, that how they make one nucleus to 4 nucleus, whether mitosis or meiosis, because that is not known yet. Whether single division and single replication of DNA and then double division, or double replication of DNA and double division, it is not yet known. So I am still interested, few more years left for my job, and I would like to work with this Amrita and Susanto. Two of them, one is working with topoisomerase, another is DNA helicase, so that we can figure out a little bit at least. And Pooja is working on apoptosis inducing factor and autophagy during encystation. So this is my little story, that what I did. During my postdoc I did with Professor John Samuelson. Currently he is in molecular cell biology department, Boston University Medical Center, Boston. Anirban was postdoc that time, Katrina was graduate student, and Lakshmi Iyer was in NCBI, who helped me to identify the transcription factor by bioinformatic way. And definitely these are the students. After I stopped postdoc, I almost stopped working by myself. So everybody contributed here. Diburupa and Arindam working on the drug activation and drug resistance part long back. Now I stopped this. All this work done by the money of IIT Kharagpur, DBT, CSIR, ICMR, and MHRD, currently MOE, the Ministry of Education, Government of India, and West Bengal Department of Science and Technology and Biotechnology, Government of West Bengal, their fund. So if there is any question, I'll be happy, or if there is any detail, like all these papers are available in net. So anything you can, whatever you remember, I don't know how much you remember after this long discussion, you just give the keyword and comma and Ghosh SK, you can find this paper in the Google. Or if you go to Google citation or Sudeep K. Ghosh Google citation, list of all my papers will be there. You
Yes,
can click
sir.
there.
So they can visit your profile and they will get all the information. And
Yeah,
your
yeah.
work is really brilliant, sir. So much things we got to know here. Many of the
So,
things we didn't know, but it was so enriching and very. I'm very excited
so
after hearing so much about the
this
work.
is also 3 cyst. Suddenly I found in this way. So
Answer the pictures, the images and the videos, they are mind blowing. I have never seen this kind of images. Thank you, sir. Sir,
Thank you, sir.
there is a question. The question is, just a minute, sir. I will just tell. Somebody has written in the chat box.
Okay, well, there are so many.
Yes, sir. So many things has been written. Yeah. There is a question from Shumanto Baski that in the intestine can tryptase degrade or damage cyst wall?
No, it is inaccessible. And tryptase, I mean, so far I know, all these proteolytic enzymes are in the stomach. Trypsin, chymotrypsin, erepsin, all are in the stomach. They don't work in intestine because there is the alkaline pH, one is the lipase, and some carbohydrate degrading enzyme which is alkaline, that can work only. But these enzymes like chymotrypsin, trypsin, erepsin, they are working only in stomach in acidic pH. So they cannot do anything to cyst wall in. But the trypsin, chymotrypsin, they may do something, some chewing, so that it is helpful for them to exist. Is the answer is okay?
Yes, sir.
Okay. Okay, so I don't know. You must be tired.
Yeah, are there
now.
any more question? You can directly ask, sir, if there is any more question.
I'll be happy to answer or explain if there is any part that is not understandable.
Sir, actually, everything was explained so well that I don't think there is anything left to understand.
No, sometimes it happens because after lunch, after listening 2 hours, 1.5 hours
No, it
seminar.
was very engaging, sir. I myself was I got so much interested in your topic. The whole time I was listening. There are many things which I couldn't understand, but overall, the thing which you tried to say was quite clear and understandable. It was very nice. The work is very interesting, sir. Yes, so sir, I don't think there are any question left. So sir, again, thank you very much from the organizing committee. It
Thank
was
you.
really, yeah, sir. It was so much engaging and the work was brilliant, sir. Thank you, sir.
Thank you.
Yes,
Okay.
sir.
All the best to all of you.
Thank you, sir.
So let me disconnect.
Yes, sir.
Okay.
So today's session is over. All the participants can leave. Again, we will meet tomorrow at in the morning, in the first half hour of our session, probably at 10:30 a.m. So till then, good night. Thank you.
Thank you, ma'am.
Thank you, ma'am. Thank you,
Thank
madam.
you. Thank